In this video, we will discuss about how this uh, purinergic receptor can also lead to immunogenicity. So, what is this purinergic receptor? It is the combination of either adenosine nucleotide or is some modified form of adenosine nucleotide such as adenosine triphosphate. So, when they are present in the extracellular matrix in the higher amount, they can trigger immune response. And in this figure, different kind of immune response are shown that is related to these uh, purinergenic receptors. So, and since we know that cancer cell arises from different kind of mutations that has occurred that leads to the uncontrolled growth rate, and that different mutations will also leads to the increase in the purine metabolism, or the purine nucleotides will increase because of the mutations occurred in the cancerous cells, and this will leads to the surrounding of these cancerous cells. This will lead to a condition where surrounding of the cancerous cells will have high amount of uh, purines, that is ATP or adenosine nucleotides. And this high amount of ATP or adenosine nucleotides, they can interact with this effector T cell receptors such as P2X, P2Y, or A2AR. And by when they interact with this kind of receptors on the effector T cells, then it will release the cytokines, and also it will proliferate the effector T cells. And simultaneously, what it will does is the it will interacts with the dendritic cells. It will interacts with the dendritic cells to it will interact or instruct dendritic cells to further proliferate the effector T cells with the antigen informations that is received from this not the antigen information not received in this case. In this case, the dendritic cells get activated by the dendritic cells will also co-stimulate the effector T cells, and this will lead to the high proliferations of the T cells. And the other immune cells that will also be activated because of the high amount of purines, that is neutrophils. Neutrophils also has this kind of receptors such as P2X, P2Y, and when these neutrophils receptors interacts with these purines, then it will leads to this chemotaxis formation such as TNF or IL-17. And both of these cytokines or chemotaxis has the immunogenic effect against the cancerous cells. And these adenosines will apart from effector T cells and neutrophils, the regulatory T cells and NK cells, regulatory T cells will also has the receptors. And when these regulatory T cells get activated, then it will lead to the release of the cytokines such as interleukin 10, TGF beta, and this will also trigger the apoptosis of these regulatory T cells. Regulatory T cells has a function to suppress the immune response. So when these high amount of adenosines are present, then it will lead to the apoptosis of these regulatory T cells, or in other terms, regulatory T cells will go down. So the suppression of the immune system or immune suppressions because of the cancerous cells will decrease. And then the other things that will happen that will promote the pro-tumorigenic effect is that these adenosines will interact with these monocytes, and that will lead to this M2 phase phenotype differentiation. And we have discussed that M2 phase phenotype differentiation will help in the pro-tumorigenic effect by suppressing the immune response. And whereas if this monocyte is differentiated to this M1 phenotype, then it will have anti-tumorigenic effect, and it will so we can say it will it will have anti-tumorigenic effect, and it will try to kill the tumor cells. So when it comes to the NK cells, there is because of the high purine metabolism, this Mica receptors get interact with this NKG2 D receptors on the immune cells, and these interactions will lead to, because of the high purine metabolism, that will lead to the overexpression of this Mica on the cancerous cells, and this mica can be detected by the NK cells through this NKG2D receptors, and when this interaction occurs, this will lead to the immune activations by the NK cells. So in this case, majority of the conditions, it will promote anti-tumorigenic effect, except few conditions such as this monocytes differentiation to M2 phase. And the other things that it will cause is that high disruptions, imbalance of the purine to pyrimidine ratio, because mutations leads to the high purine formations. So because of the disbalance of this purine to pyrimidine ratio, this will leads to the breakage of the cell membrane of the cancerous cells, and this will cause this purines in the extracellular matrix of the cancerous cells, and this will increase immunogenicity and the sensitivity to immune checkpoint blockade, as uh, it will decrease the amount of regulatory T cells that contains the immune checkpoint ligands such as CTL4, PD1. So that will be reduced whenever this high amount of purines are there in the extracellular matrix around the cancerous cells. The next thing is similar to this purigenic immune response through this purigenic receptors is about this microbes that will release nucleoside analogs to regulate immunity, or microbes will release nucleoside that is similar to these purines. So and those are known as inosines, and these inosines will also be able to interact with the receptors, this A2AR, that are present on the affected T cells, and that will trigger immune response. And the other way by which this microbe trigger immune response is through this cytokines such as CDI AMP, and it will help the monocyte differentiation to this macrophages, or we can say that M1 phase, and or what it will does, and simultaneously this monocyte will also release this also release this type 1 interferon, and that will activate the NK cells. NK cells and dendritic cells are in a cascade effect, that it will they are in a feedback loop, we can say that. Those, when the NK cells get activated, it will release this CCL5, XCL1. It will upregulate this CCL5 and XCL1 are the cell surface receptors on the NK cells. They will be upregulated, and it will lead to the high amount of dendritic cell formations. And when there are less amount of these microbes are there, then dendritic cells will release IL-15, that will suppress the or that will decrease or suppress, or we can say deactivate the NK cells. So thereby these dendritic cells and NK cells will act themselves in a feedback loop themselves as a feedback loop, so that they can control the overexpression of NK cells or the dendritic cells. So I hope you got an idea how these purigenic receptors and immunogenicity are related. And if you need to understand this topic in detail, that is how this imbalance of purines to pyrimidine ratios might trigger immune response, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.